Good morning, everyone. As Naboisha just said, today I'm happy to be talking to you about a collaboration we have with Anna Pyle's lab at Yale, where we've been looking at this modified DNA aptamer that binds a protein called RIG-I. And RIG-I stands for retinoic acid inducible gene. It's a cytosolic pattern recognition protein that when it binds viral RNA activates downstream production of interferon. It's a large multi-domain protein that recognizes short, duplexed, blunt-ended, 5' tri or diphosphorylated positive strand viral RNA, such as that found in SARS-CoV-2. Agonists of RIG-I are currently of interest because it's known that currently of interest in cancer immunotherapy because it's known that signaling of RIG-I can trigger cell death. On the opposite end, antagonists of RIG-I are also of interest because aberrant signaling of the protein is linked to autoimmune diseases and autoinflammatory diseases. And so Anna's lab has been studying the structural requirements of RIG-I RNA binding for quite some time. And they were interested in developing a RIG-I antagonist. And they had a nucleic acid antagonist that they just weren't happy with. It had pretty low affinity. And she wanted to know if we were interested in working with her to develop a modified DNA antagonist. And we'd worked with Anna's lab in the past where they solved the structure of one of our reagents bound to a completely different protein. And we were hoping that we could get another structure out of this collaboration to help us further understand how our reagents are folding and interacting with their targets. So we started this project first by doing a selects to RIG-I. At Soma we can identify high affinity target specific aptamers using modified nucleotide DNA libraries and selects. And so for this selects, we had 3 targets or different constructs of RIG-I. We had the full length protein, the RIG-I protein lacking these CARDS domains, as well as the small C-terminal domain. And it's this C-terminal domain that I should mention is responsible for the high affinity interaction with the RNA. And so for each of these 3 targets, we did a selection using 3 different modified nucleotide libraries. We have NAPDU, TRPDU, and PPDU. And so here I'm just showing you a standard round of selects. That's our standard procedure. And I just want to point out that we have this step here where we have a polyanionic competition. And that allows us to select for reagents with slow off rates. And because of that, we call our aptamers that come out of selects SOMAmers, which stands for slow off rate modified aptamers. And so we use modified nucleotides in selects because we found they give us the greatest success rate. And we know that these modifications are critical for folding of our SOMAmers. As you can see, they form these unique structural motifs within the folded aptamer. And we also know that they are crucial for interacting with their protein targets. So following the completion of 9 rounds of selects, the pools were sequenced and we ordered 140 sequences to be synthesized as 50-mer synthetics to be screened to determine their binding affinity to their targets. And they were screened in a gel shift assay. And as you can see, we had many high affinity binding sequences across all of the modifications and all of the targets. But we decided to first focus on the RIG-I CTD, as this made the most sense as a target for finding an antagonist. And we also knew it was a good target for crystallography studies. So we selected six sequences, six of these high affinity sequences that encompassed all three mods for truncation optimization. So following truncation, we had sequences that ranged in length from 25 to 41 nucleotides. And we sent these six sequences to Anna's lab where they began crystallization studies. And they were able to solve the crystal structure of one of them bound to Rigai CTD. And we call this somomer in this structure SL1090. And it's a 41 nucleotide somomer, and it has 16 of these PPDU modifications that you can see here in the sequence in orange. And we were really excited that we had this modification in this crystal structure because it was the first time we'd seen it in a structure. And this modification has this long linker. And we were thinking, and we always kind of assumed that this long linker meant that this mod would find deep pockets, deep hydrophobic pockets that it could bind to on the protein surface. So before I tell you what the mods were actually doing in this structure, I just want to share some statistics. You can see that this somomer binds Rigai CTD with very high affinity, 19 picomolar. It also has maintains good affinity to the full-length protein. It was a high resolution structure at 1.9 angstroms. And although it was the longest somomer sequence that we'd crystallized, it actually had one of the smallest interface areas at 862 square angstroms. But that really high affinity in that small interface area means that it has excellent ligand efficiency at 0.286 kcal per mole per contact atom. And interestingly, it's a really compact molecule having a surface area to volume ratio of 0.407. And as far as our other somomer structures go, this one actually has quite a bit on the low end for shape complementarity at 0.587. So we have 5 additional somomer structures, somomers bound to their protein structures. And all of these somomers structures are really unique and completely unpredictable. But I think this one might be the strangest. And the PPDU mods in this structure are doing exactly the opposite of what we thought they would. So only 2 of those 16 modifications are contacting the protein. The rest are all directed inward where they form a massive hydrophobic core. And the structure itself can be divided into 3 parts. There's the terminal stem, which just has a few Watson-Crick base pairs. And then there is this triplex region where the bases are hydrogen bonding to form base triples. And the mods associated with those bases are forming that hydrophobic core that I just mentioned. Also in the triplex region, we see yet another example of this zipper motif. And so this is when we have mods originating, or modified nucleotides originating from opposite strands, stacking in such a way that they replicate base stacking. And in this example, it's not quite as planar as we've seen with other modifications. And I believe that's because the linker is so long that it has to kink a bit in order to allow the base mod stacking. And then the last region is what we call the nested loops. And this is a motif that's completely unique to this structure. It involves 5 different loops that are color-coded here, all coming together to interact through base stacking and base pairing among the various loops. We also looked at the nucleotide conformations in this structure. And as we all know, nucleic acids have 6 rotatable bonds in their backbone. And Anna's lab has come up with a really clever way to analyze these nucleotide conformations where they reduce those 6 rotatable bonds to 2 pseudo torsion angles called eta and theta. And so it allows you to do an analysis similar to phi-psi angle analysis with a protein structure, if anyone's familiar with that. And so in this structure, we found that 27 of 39 residues fall outside the normal metrics for helical geometry. And if you had those statistics in a protein structure, someone would tell you that your structure was complete garbage. But in this case, it's just a testament to how flexible that nucleic acid backbone really is and how the molecule can adapt to accommodate these PPDU modifications. And then on the right here, I'm showing you the Soma structure color-coded by B factor, which is the temperature factor. And so the way to look at this is regions that are color-coded blue are rigid and regions that are red are more dynamic. And so in this case, the blue regions that you're seeing here are where the somomer interacts with the protein. And so now if we turn our attention to the protein somomer interface, we can see that it's really dominated by polar interactions. And this is quite a departure from what we've seen with all our other structures where the modifications are really highly represented at the interface. Here in panel B, you can see one of the two PPDU modifications interacting with the protein, where it's stacked here with the methylene portion of this lysine residue. And then in panels C, D, and E, you can really see this network of hydrogen bonding with the protein, primarily with the somomer backbone, as well as here with one base involved. And all of these interactions are mediated by water molecules, or in this case, a magnesium ion. But what's really cool is actually if we look a little bit closer at the interface and we compare how the somomer is binding with how 5' phosphorylated double-stranded RNA is binding, we can see that the somomer really mimics how the RNA interacts. And so just looking at these orange ribbon diagrams of the somomer and the RNA, you can see the structures are completely different. But then looking at how they're interacting, you can see they're using very similar strategies to bind the protein, even binding the same exact residues on the protein. And so we have done some preliminary experiments that show that SL1090 is capable of inhibiting signaling of RIG-I, but we need to do some further analysis and add additional controls to really be sure about those results. And so now for the last few slides, I just want to completely change gears here and tell you a little bit about this project we've been working on for the last 2 years in collaboration with William James's lab here at Oxford. And this is where we've been developing a somomer to SARS-CoV-2 spike protein that has antiviral activity. And so this somomer is called SL1111_20, and it's a 29mer. And this reagent actually has two modifications, one on C, which is NAFDC, and one on U, which is TRDU. And here I'm just showing you the binding affinities to recombinant spike proteins, the wild type monomer and wild type spike trimer, as well as trimer versions of all of the current variants of concern. And you can see that there's no loss in binding affinity to all of these variants. And what's more exciting is that there's actually, this reagent is capable of neutralizing virus. And we see actually also no loss in antiviral activity among all the variants that we've tested. So we were wondering, how is this reagent able to bind the spike protein that has all of these mutations? We don't have a crystal structure, unfortunately, of this reagent bound to spike, but we do know that it targets the receptor binding domain of the protein. And there are lots of antibodies that bind spike RBD, and they are classified, they fall into one of 4 classifications, depending on where on the protein they bind. So the class 1 and 2 antibodies that are shown up here, their binding epitope overlaps that of the ACE2 receptor. And these are the most potent antibodies, but they're also completely ineffective against all of the variants. And down here, away from the ACE2 binding site, are the class 3 and 4 antibodies. These aren't as potent, but they are effective against the variants. And so one of William's colleagues here at Oxford, Jack Tan, did this assay where he took a panel of antibodies that represent all 4 classes, and he used our reagent and determined, and he did an ELISA competition assay to determine which antibodies are blocked from binding spike when our reagent is present. And so we found that there are specific class 4 and class 3 antibodies that our reagent blocks from binding, as well as to a much lesser extent, this class 2 antibody. And so this means when our reagent is bound to spike, these antibodies cannot bind. Therefore, they have a similar or overlapping binding epitope on spike. And so we used that panel of antibodies. They all have structures. And we looked at, we mapped the binding sites of all of the antibodies. And then we asked which residues on spike are unique and only found in the antibodies that were blocked by our reagent. And so those are mapped here onto the sequence of the spike RBD. We also did a hydrogen deuterium exchange mass spec experiment to help us further identify regions on spike RBD that our reagent could be binding. And so those regions are highlighted here in green, and we could see we had pretty good agreement between these 2 types of analysis. And below the sequence of spike, I'm showing you where there are mutations in the various variants. And you can see that it looks like our reagent is binding fairly conserved regions where there aren't too many mutations, or if there is a mutation, it's a mutation in our favor where a residue is mutated to a positively charged amino acid. And so here I'm showing you these residues mapped onto the spike RBD structure. And we don't yet really understand how our Somamer is binding these discontinuous regions of the protein, but we're hoping to have a structure of this pretty soon that will help us understand these results. And with that, I would really like to thank all the people we've worked with at Yale. Jiaming Ren was the postdoc who solved the structure of Rigai bound to Somamer in the lab of Anna Pyle. And here at Oxford, we've been working with William James, Jack Tan, Seilu, and Adam Harding did a lot of the neutralization assays that I showed. And then, of course, my colleagues at SomaLogic, I'd like to thank Naboisha and my boss, Dan Schneider, as well as Jeff Carter and his whole group in the synthetic chemistry lab.